Hello, good morning. Welcome to our first Greenstone webinar. I'm going to wait just another minute to let more attendees log on, and then we'll get started. Okay, I'll go ahead and get started. Welcome to our first Greenstone Biosciences sponsored webinar. The title of the webinar for today is NAMs, which are New Approach Methods. NAMs hold the promise to bring human biology to the earliest stages of drug discovery programs using patient-induced pluripotent stem cells. By way of introduction, my name is Dr. Todd Herron. I am a PhD scientist by training. The majority of my training is in molecular and cellular cardiology, and I also have an extensive training and background in using stem cells for biomedical research. In fact, I have 15 years of experience in the iPSC field developing new approach methods, or NAMs, specifically for cardiac safety. For the last 20 years, I served as a faculty member at the University of Michigan and was the founding director of the University of Michigan Cardiovascular Regeneration Core Laboratory, where we derived new iPSCs and also differentiated those into various different cardiovascular cell types. Currently, and since February of 2025, I'm the director of the HIPSC Biobank here at Greenstone Biosciences. And I'm very proud and fortunate to be a member of the Health and Environmental Sciences Institute Cardiac Safety Committee, specifically on the iPSC cardiomyocyte validation team from 2016 to now. And I'm very proud that being a member of this team, I've helped to develop NAMs that are changing the way regulatory science is performed at the FDA and the way that new drugs are screened for cardiac safety. To give and provide a little bit of perspective on where I'm broadcasting this Zoom webinar from, our company, Greenstone Biosciences, is located at Stanford Research Park in Palo Alto, California, on the west coast of the United States. And specifically, our labs and offices are housed in the Alexandria Center for Life Science at Stanford Research Park. To provide a little bit of background and perspective for the webinar content for today, I've included an agenda. The primary goal of our webinar today is to discuss new approach methods, or NAMs, and how this is creating a new paradigm to significantly advance drug discovery and drug development projects. I'll provide a little bit of background on U.S. federal legislation and how the FDA and National Institutes of Health are calling for NAMs to replace the use of animal testing in many facets of biomedical research, as well as drug discovery and drug development. I'll provide some background on Greenstone Biosciences and focus on how our company is rooted in 20 years of NAM development by its co-founder out of the School of Medicine at Stanford University. Then I'll introduce the Greenstone Biosciences NAMs, which are primarily predicated on our extensive HIPSC biobank, which contains over 2,000 unique patient cell lines. And at the end of this webinar, I will present some of the greenstone NAMs, some of our products, our cell products, including clinical trials in a dish based on iPSCs. I'll present our differentiated human cells products. I'll present our 3D organoids for different organ systems, including cardiac, the GI system, the liver, and vascularization of those 3D organoids. And I will also show how the greenstone NAMs can be utilized for high throughput screening for drug discovery and drug development projects. So to provide a bit of background on the federal level, recently on June 10th, 2025, in the journal JAMA, the new FDA director published an article titled Priorities for a New FDA. And in this viewpoint article, the new director posed his first question in the article is, why does it take more than 10 years for a new drug to come to market? Further, in the viewpoint, the new director addresses a problem that has contributed to the long duration of drug discovery and drug development before a drug can come to the market. And that problem outlined in this viewpoint is that animal testing is expensive, is time consuming, and does not accurately represent human responses. As a result of the use of animal testing and drug discovery results in wasted time, wasted resources, and contributes to the high cost of new drugs and new therapy development. The director also lays out a solution to this problem of the current long duration of drug discovery and drug development, and that is a roadmap to reduce the use of animal testing for drug discovery and toxicity testing. This is some information showing that animal tests fail traditionally to predict safety in humans for new medications. So currently, in the traditional paradigm of preclinical testing, the FDA's drug approval process depends largely on animal tests. Many animals are utilized in these preclinical tests. There's lethal dose 50 testing that is done. And once a drug compound is deemed safe in animals, that drug compound then progresses to clinical testing. And unfortunately, in clinical testing is where some of the toxicities that were missed in animals become apparent. And here are two different examples. And example one, in France in 2016, an experimental painkiller trial led to one death and landed others in the hospital with brain damage. This particular drug in preclinical testing successfully passed safety screening in rats, in mice, monkeys, and in dogs, none of which caught the human toxicity, which was apparent in the clinical testing. Another example from Seattle in 2016, a cancer drug trial after being tested in animals and was given to humans was found to cause cardiac arrest and death, unfortunately, in some patients. Again, this drug in the preclinical setting was tested successfully and passed toxicity safety screening in mice as well as in dog models. So this led to a quotation from the former NIH director, Francis Collins, that the use of animals for therapeutic development is time-consuming, costly, and may not accurately predict efficacy in humans. The solution to this problem of animal testing failing to catch human toxicity is to bring human relevant biology earlier to the drug discovery process using new alternative methods, or NAMs. And here I'm presenting the use of iPSCs as a primary NAM for drug discovery and drug toxicity testing. And it relies on the revolutionary discovery from 2006 from Dr. Yamanaka in Kyoto, and that was the invention of iPSCs, which are induced pluripotent stem cells that are derived from patient somatic cells, which may be a blood draw or a skin sample biopsy. And once the iPSCs are established in this diagram, you can see that the iPSCs can give rise to many different cell types. In fact, all cell types of the human body can be reproduced and recapitulated in vitro using iPSCs. And as the toolkit of iPSCs for in vitro studies has grown, now we can make very complex in vitro systems, including organoids and engineered heart tissues that can be utilized to recapitulate, in this example, the function and the structure and the response of the human heart to different drugs to test for drug toxicity as well as drug efficacy. And this new paradigm based on iPSCs has led to an idea and a method called clinical trial in a dish, where various participants now, where iPSCs have been made for specific patients, these patients can be represented in a clinical trial in vitro. Remarkably, iPSC-derived cells recapitulate the genetics and the functional and structural phenotypes of the patient that the cells are derived from. In this new paradigm for clinical trials in a dish, here the example utilizes iPSC cardiac organoids. It's possible to test new medications and to determine which patients respond to medications, which patients have toxic effects or toxic responses to specific medication, and to determine responders from non-responders. This paradigm is promising to increase the success rate of drug development and the delivery of new drugs and new medications to the market to patients who need those new therapies and those new treatments. As the iPSC toolkit has expanded, now there are becoming more complex in vitro model systems called MPS, or microphysiological systems, where multiple cell types and multiple organ systems can be studied at once in order to determine toxicity with better fidelity and higher throughput. All of this information that is being learned in vitro through clinical trials in a dish can then be fed into artificial intelligence and machine learning algorithms in order to better plan and predict outcomes in the clinical trial where the medications are actually given to patients in a hospital setting. So here we're reviewing how patient-specific iPSCs represent a very significant NAM, a new approach method. Utilizing iPSCs, any patient and any patient population can be represented in vitro to gather in vitro information prior to administering any medication to a human. This allows for testing new medicines on target populations in vitro as well for better efficacy screening and efficacy testing. The clinical trials in a dish paradigm is going to inform and make more successful future human trials. Overall, the use of iPSC NAMs in clinical trial in a dish is expected to reduce the cost and the time of drug development while increasing the success rate for drug development and delivery of new medications to the clinic to treat patients for a myriad of diseases. To further promote and advance the utilization of NAMs for drug development and drug discovery, recent U.S. bipartisan-backed legislation from Congress has been passed to make drug discovery and toxicity screening more efficient. And this U.S. legislation is called the FDA Modernization Act 2.0 or 3.0 and has impact on the drug development landscape. In 2022, There was the first passage of the FDA Modernization Act 2.0. The federal legislation amends the longstanding Federal Food, Drug, and Cosmetic Act. And the language in this legislation formally allows for the use of alternatives to animal testing in drug development projects. This has enabled a shift of focus away from animal models and animal testing in drug discovery and development towards human-based testing and the use of NAMs, new approach methods. The goal of this legislation is to make faster and safer drug development processes for patients and for drug makers. Since 2022, there's been a lot of excitement around the FDA Modernization Act. However, there hasn't been a lot of changes in the regulatory landscape at the FDA. This spurred the U.S. Senate in 2025 to pass the FDA Modernization Act 3.0, which is now awaiting House approval before passing. In the FDA Modernization Act 3.0, imposes a 12-month deadline on the FDA to amend its regulatory paths to now include NAMs in place of animal testing for drug discovery and drug toxicity screening. The FDA Modernization Act 2.0 and 3.0 is having significant impact not only on the FDA, but also on federal funding and policy at the National Institutes of Health. Recently, the NIH has announced a priority to prioritize human-based research technologies in place of animal testing. Specifically, the NIH director has announced the establishment of the Office of Research Innovation, Validation, and Application, an acronym ORIVA, within the NIH's Office of the Director. The goal of ORIVA will be to expand funding and training in non-animal approaches and awareness of their value in translational success for drug development and drug discovery projects. Importantly, the NIH director has pointed out and has made a point to develop more infrastructure in the NIH for non-animal approaches to be expanded. Importantly, regarding NIH federal funding, there's going to be a directive for grant review staff to participate in mitigation training to address any possible bias towards animal studies and to integrate experts on NAMs into study sections. The FDA Modernization Act will have great impact on drug discovery and drug development. And this diagram depicts the traditional drug discovery and drug development timeline as it's been known to the field, from the pre-discovery phase through the preclinical stage, focusing on animal studies. The preclinical stage for a compound on average requires 3 to 6 years of testing and 3 to 6 years of animal studies before a compound is deemed safe and effective to pursue clinical trials. The clinical trials, which traditionally is the first time that a new molecule and a new therapy touches human biology or is tested in humans, is 6 years, 3 to 6 years after the pre-discovery phase. And in this phase of the clinical trials, many drugs fail due to efficacy or toxicities that were missed in the animal studies. So what NAMs promise to do for the drug discovery and development timeline is to bring human biology closer to the preclinical stage. So utilizing iPSCs as NAMs, now it will be possible for regulators and for drug makers to bring human biology in the form of NAMs to the preclinical stage in order to make drug development and drug discovery a more efficient process that doesn't require so much time in animal testing before it's first tested in humans. So, this new legislation from the US government creates new opportunities. One opportunity is accelerated drug discovery and drug development times when using iPSC focused new approach methods, or NAMs. And this also promises to reduce the costs associated with drug discovery and drug development. Greenstone Biosciences, founded by Joseph Wu and Jade Chow, is a biotechnology company that's really rooted in NAM's development. Rooted in 2 decades of NAM development using iPSC NAMs that have been developed and tested and validated by Dr. Wu's laboratory at Stanford University. Greenstone Biosciences, in addition to providing NAMs, also has a drug discovery pipeline and has several molecules in clinical trials. One of our primary assets at Greenstone Biosciences is our extensive iPSC biobank, which contains over 2,000 ready-to-use patient cell lines that can be thawed, expanded, and utilized for human NAM production and testing for drug discovery and drug development projects. Dr. Wu's laboratory over the last 2 decades has published many commentaries as well as original research articles in high profile and high impact journals uh, with titles discussing induced pluripotent stem cell technology and perspectives on decades of progress. Uh, Articles focused on progress of multicellular human cardiac organoids for clinical applications. Recently, in conjunction with other researchers at Stanford University, there have been reports on the use of clinical trials in a dish for cardiovascular medicine and cardiovascular drug development. And recently in 2024, in collaboration with FDA regulators, Dr. Wu's group published a paper entitled Tackling the Challenges of New Approach Methods for Predicting Drug Effects from Model Systems. And in a very exciting recent publication in 2025 that just came out in the last month, Dr. Wu's laboratory developed gastruloids, which enable modeling of the earliest stages of human cardiac and hepatic vascularization. So now I'm going to present some Greenstone Biosciences biobank services and want to bring to your attention that we can be your partner in advancing drug discovery using iPSC-derived new approach methods, or iPSC-derived NAMs, to promote drug discovery and drug development projects. So our biobank at Greenstone Biosciences is cutting edge, and we focus on harnessing the power of iPSCs, or induced pluripotent stem cell technology, for drug discovery and human-based biology at the early stages of drug discovery. Our biobank is able to differentiate different cell types, virtually any cell type of the human body that can make a meaningful and a useful NAM. And we can use these cells for disease modeling, for high throughput screening, and for toxicity testing. Our capabilities enable us to conduct very early stage safety assessments, which results in significantly de-risking drug discovery projects at the earliest stages of drug discovery projects. And at Greenstone, we are dedicated to pushing the boundaries of science to transform patient outcomes. This is a look at our approach for genetic and pharmacogenomic studies, utilizing NAMs to overcome the limitations of traditional animal and in vitro model systems. So, in vitro model systems, such as using immortalized cells in vitro, do not accurately represent the large genetic variability of the human population. So, these assays do not accurately predict and represent the diverse genetic makeup of the human population. Additionally, in using inbred mice in many biomedical studies to study the effects of a specific drug, there's very little genetic variability in these inbred mouse strains. And so these also are models using inbred mouse strains that do not accurately represent or recapitulate the very diverse genetic makeup of the human population. So our solution is to go straight to the human cells of many different patients that can be represented in vitro to be able to recapitulate and to be able to study and understand the very large genetic variation and the genetic variations that lead to differential responses for specific drug medications on specific patients, which can be divided into different populations to be able to get a better handle on how a specific drug response comes out of a specific patient and a specific patient population. Here's an example of the workflow that we utilize at Greenstone. The workflow, we can test basically any modality of therapeutic, ranging from vaccines to small molecules, genetic-based therapies such as antisense oligos. We can perform studies on antibody-based drugs. We can study the effects of other RNA-based therapeutics and also peptide-based drugs on human NAMs in vitro, performing large-scale high-throughput screening for in vitro candidate drug testing. Utilizing this approach, we then can proceed to a small-scale animal study to determine the phosphokinetics, or pharmacokinetics, and dynamics in animals. Alternatively, where animal studies are relevant, for example, in the case of antibody drugs that are species specific, we can then fast track straight to clinical trials in a dish where the animal studies would produce irrelevant data. So in the clinical trials in a dish, utilizing cells as monolayers or more complex three-dimensional organoids of any organ system of the human body, or microphysiological systems with microfluidics, we can then test these drug medications on various patient populations in vitro to determine which patient populations respond well or which patient populations may experience an unforeseen toxicity in vitro before proceeding to clinical trials. The information gleaned from the clinical trials in a dish can then be wrapped up and utilized in machine learning and artificial intelligence algorithms in order to predict ideal clinical trials designs, which would then allow for proceeding to informed clinical trials where the clinical trial can be formed and planned based on a wealth of human relevant biological data collected in vitro through the clinical trials in a dish approach. So here I want to introduce the Greenstone iPSC Biobank. We're very proud of our iPSC Biobank. To our knowledge, it's the largest iPSC biobank in the world with greater than 2,000 patients represented in our biobank. And we utilize this biobank for accelerated drug discovery programs. The 2,000 plus patient cell lines come from a diverse patient population, and we also have some cell lines where patient mutations have been gene edited to create isogenic cell lines. And we also have many patients from rare diseases that are represented in our biobank. Importantly, our biobank cell lines are linked to clinical data and clinical information of the patient from which the iPSCs are derived to provide a comprehensive patient-specific insight into any clinical trial and a dish data that we discover in our clinical trials in a dish. On the right, you can see a pie chart that shows the disease representation of patients in our biobank. The largest proportion of cell types in our biobank are from cardiovascular patients. These are patients largely with inherited cardiac arrhythmias or inherited cardiomyopathies. We also have neurological diseases, neurological patients represented in our biobank. There are gastroenterology patients in our biobank. There are also oncology patients, patients diagnosed with cancer that we have iPSC lines for in our iPSC biobank. And for musculoskeletal diseases, we have iPSC lines in patients represented in our biobank that have musculoskeletal disease. And there's many other diseases not listed here that are also represented in our biobank, including many rare diseases. So utilizing our biobank at Greenstone, we can deliver end-to-end iPSC solutions from cell sourcing to cell screening and NAM development and NAM testing in vitro. And our goal is to utilize our biobank in order to accelerate innovation and to accelerate the de-risking of drug development. We also have capabilities to perform multi-omics analysis, and our rich and diverse biobank allows multi-omic studies across diverse populations to understand how different populations respond genetically and transcriptomically to different drug therapies and drug treatments, which allows us to have a very intimate understanding of the genetic impact of drugs on the responses of patient cells. So here I want to provide one way that companies that are fostering drug discovery and drug development projects might partner with Greenstone for the development of iPSC NAMs, and that is for the creation of clinical trials in a dish. So if you partner with Greenstone Biosciences to de-risk your drug discovery compound, your lead compounds, the first step is that the customer can choose the patient cell lines to be tested. And the customer can choose from our vast array of 2,000 patient cell lines in order to represent their patient population of interest or to represent a diverse population in a clinical trial in a dish. After selecting your patient cohort of interest, then the scientists here at Greenstone will retrieve those iPSC lines from the specific patients. We then thaw and expand the patient iPSCs, making sure that those cells are of very high quality, very high purity of pluripotent stem cells. Once we have a highly pure and highly differentiation potential iPSCs from those specific patients, we then can differentiate those stem cells to the cell type of interest. And in this example, I'm showing differentiation to cardiomyocytes, which can be tested as two-dimensional monolayers, which you can see in the top portion of this clinical trial in a dish. Or alternatively, we can also differentiate those cells into three-dimensional cardiac organoids that can also be tested in high-throughput screening systems to determine drug impact on either two-dimensional monolayers of cells or three-dimensional cardiac organoids. Here, this is a table that provides some examples of cardiac diseases and the genetic variants that have been identified in those specific patients' cell lines. We have Different cardiovascular diseases represented, such as atrial fibrillation, Brugada syndrome, dilated cardiomyopathy, hypertrophic cardiomyopathy, long QT syndrome, and left ventricular non-compaction. And for each of the cell lines that are represented in our biobank, we also have DNA sequencing information, and we know which variants are mutated, which genes are associated with each case of inherited cardiomyopathy. In this movie, I'm showing an example of 3D iPSC derived cardiac organoids that were generated here in Greenstone Laboratories utilizing healthy control patient cells. We can generate very large batches of three-dimensional organoids. Here you can see those organoids contracting spontaneously. Each organoid essentially has its own pacemaker and can be used to study contractile function of the organoids from any patient. For example, patients with inherited dilated cardiomyopathy might have contractile impairments that we can detect utilizing these three-dimensional cardiac organoid NAMs in vitro. We also utilize fluorescent dyes. Here on the right, you can see the spontaneous electrophysiology by measuring the intracellular calcium transient that triggers each contraction of each organoid. You can see each organoid is working independently. That's because each organoid has its own pacemaker and functions as its own independent cardiac organoid. At Greenstone, we also have high throughput screening machines and instrumentation where we can utilize high throughput screening plates, predominantly 96 well plates or 384 well plates. And in these systems, we can measure electrophysiology, to determine any arrhythmia phenotype or any arrhythmia response of specific patients' cells or cardiac organoids to different medications. And so our instrumentation allows us to record functional phenotypes, including electrophysiology screening, toxicity screening, transcriptomics, genomics, and as well as proteomics. Additionally, in our biobank, we also have many rare diseases represented from patients diagnosed with rare diseases such as Loeys-Dietz syndrome, Lynch syndrome, Marfan syndrome, Duchenne's muscular dystrophy, myotonic dystrophy type 1, and TTR cardiac amyloidosis. Similarly, for these patients with rare diseases, we also have genetic information where we can describe and define the specific mutation, for example, of the DMD gene for Duchenne's muscular dystrophy. We know which patients have which mutations. This is also a great strength of our biobank, is the ability to model the same disease caused by a diversity of mutations that lead to the same disease. This is an example of a clinical trial in a dish that can be performed if partnering with Greenstone Biosciences. So in this example, in a 96 well dish, different patient populations are represented in the same 96 well plate. Here, each patient is represented by a different color, or the well is colored a different color based on the patient identity. Here, the blue is for GSB cell line 958, which is from an African American patient, a female patient. We also can include GSB line 32, which is derived from a patient of East Asian descent, a male patient. The yellow is GSB line 15. This is a Caucasian male patient. We also can represent in green a Hispanic patient, GSB line 56, a female Hispanic patient. The dark blue is showing GSB cell line L2300. This is a patient of South Asian descent, a female patient. And finally, the purple is GSB cell line L8, which is an African American patient, a male patient. So in this way, in a single 96 well dish, here we have represented 6 different unique patients where we can give the same medication to determine if there's any population specific adverse or beneficial response to a specific medication. And in this particular clinical trial in a dish, we designed it to compare a control media to a metabolic stress media in order to an attempt to model heart failure in a dish so that we could compare patients' healthy control cells, for example, GSB line 958. We can compare this patient's healthy control cells to the same patient's cells that are challenged metabolically to induce a metabolic syndrome that would lead to heart failure. So this allows for isogenic disease modeling of acquired diseases beyond genetically induced diseases. Another way to partner with Greenstone Biosciences for development of iPSC NAMs is that the customer may may provide or may already have a cohort of iPSC cells or genetically engineered cells that they would like to have Greenstone scientists expand, differentiate, and then plate into high throughput screening plates as either 2D monolayers or as three-dimensional cardiac organoids. So in this paradigm, Greenstone Biosciences can provide custom differentiation services of any cell type. This slide represents some of the cell types that we generate in our Greenstone Biosciences laboratories. Those human cell types derived from iPSCs can include epithelial cells, fibroblasts to study the structural cells of the body and cells that contribute to fibrosis. We can generate pancreatic alpha and beta cells, which are cells that contribute to blood sugar regulation in patients. We can generate hepatic or liver cells for study of hepatic toxicity. We can generate also smooth muscle cells, skeletal muscle cells to study diseases such as Duchenne's muscular dystrophy, to study new gene therapies for muscular dystrophy or new small molecule therapies for muscular dystrophy. We also can generate neurons to study neuronal activity, which can be important for studying and determining neural toxicity. We also can generate enterocytes, which enables study of the gut of diseases such as Crohn's disease or celiac disease. As I've shown before, we can generate cardiomyocytes. We can generate parasites and endothelial cells. And for studies of inflammation, we can also generate immune cells to study how these cells contribute to inflammatory processes in specific cell types. Beyond generating single cells and monolayers of cells, at Greenstone Biosciences, we also have the capacity to generate three-dimensional organoids of many different organ types. I'll show data and one new capability is to generate gut organoids. We can generate fibrotic organoids by co-culturing different cell types into fibrotic organoids. For example, we can study liver fibrosis by co-culturing hepatic cells and fibroblasts into an organoid to study liver fibrosis, or we can mix together cardiomyocytes with fibroblasts to study fibrotic cardiac organoids. We can make three-dimensional islets for study of insulin secretion for metabolic diseases such as diabetes. We make liver organoids, and based on recent publication from our co-founder, Dr. Wu's laboratory, we can also induce vasculature into these organoids, which has been a long-standing challenge in organoid development, is to be able to deliver nutrients and through vasculature to the center and the core of these organoids so that they don't become necrotic or hypoxic in the center. We also can make engineered heart tissues for contractile function measurements, as well as cardiac organoids, and we can make brain organoids for the study of neurotoxicity and neurodevelopment processes. At Greenstone Biosciences, we're always innovating and expanding our capabilities. And this slide shows a new capability in our biobank labs, and that is the gut in a dish model. So we have experts in our laboratory that can differentiate iPSCs, first to human definitive endoderm in the first step towards gut in a dish production. Uh, the next step is gut tube morphogenesis, where the monolayer of cells begins to fold and begins to form gut organoids that become beautiful 3D spheroids over time. And the end result is to have human intestinal organoids that can be used for the study of different diseases such as Crohn's disease, celiac disease, uh, intestinal cancers, or infections in the gut that can be induced by bacterial infection, such as E. coli, and also how viruses such as COVID-19 can impact on the human intestine. At the bottom, you can see real world pictures of the gut tube morphogenesis stage, where you can see gut organoids just beginning to form. You can see the development of beautiful 3D spheroids in vitro in the monolayers, and with maturation, the development of human intestinal organoids used for downstream drug discovery and drug development purposes. So in this slide, I just want to briefly summarize our Greenstone Biobank products. They're in the form of differentiated cell types from iPSCs. And the first category are cells that are produced and that are cryopreserved and can be thawed and used for the development of NAMs right away. We have many healthy control cell lines and some disease cell lines that are pre-made and can be utilized right away, including cardiomyocytes for drug screening and disease modeling. Also can be used for regenerative medicine research. We have cardiac fibroblasts, which are cells that are important for cardiac tissue repair and fibrosis studies. We have endothelial cells, which can be utilized for study of angiogenesis and vascular research. Additionally, we have smooth muscle cells, which can be used and are important for vascular function and studies of atherosclerosis. And we can make parasites, which are important for microvascular stability and barrier functionality. In the second category, these are more complex models and panels. One is an iPSC diversity panel that can be utilized for personalized research. As I showed in the previous slides, another complex model available through Greenstone is a clinical trial in a dish where a cohort of patients can be selected that is relevant to a drug in development for efficacy for a specific population, and also can be used as a valuable tool for detecting population-specific toxicities in vitro prior to administering any drugs to any patients in a clinical trial. We also offer differentiated cell kits that can be used as toxicity assays, as assay-ready plates, and also as three-dimensional organoid models. And finally, in the third panel, we also have many bespoke services. These services can include electrophysiology screening using iPSC-based NAMs. And of course, if you do not see your NAM cells or disease model of interest, of course, we can build it with you to make a meaningful NAM for your drug discovery project. Our biobank is also always expanding our services. One service that we've expanded into is to provide gene edited iPSC lines and other cell types. Our gene editing services rely on CRISPR technology to correct mutations or to introduce mutations into control cell lines for study of genetic-based therapies to determine how a genetic-based therapy, for example, might read over a missense mutation or read through a truncating mutation for a specific gene and a specific protein. We're also always developing NAMs. This is an exciting collaboration that Greenstone has with the Health and Environmental Sciences Institute, is working with the Cardiac Safety Committee on iPSC CM uses, and we're very excited to participate in an upcoming atrial cardiomyocyte specific validation study. For this study, Greenstone Biosciences can supply atrial cardiomyocytes for study of anti-atrial fibrillation drug development, utilizing genetically induced forms of atrial fibrillation. We can also recreate atrial fibrillation in a dish assays to enable study of antiarrhythmic screening. And the rationale for this study and this collaboration with the HESI committee is that atrial fibrillation is the most common arrhythmia in the United States with a high unmet need. At Greenstone, we're very well equipped and we have state-of-the-art facilities, including tissue culture suites, high content imagers, and high frame rate optical mapping systems for cardiotoxicity and neuronal toxicity studies. We also have very state-of-the-art equipment for molecular biology to perform DNA sequencing and transcriptomic analysis through RNA-seq. We also can perform mass spec analysis. We have microelectrode arrays for further electrophysiology analysis, and we can perform and routinely do flow cytometry and cell sorting as part of our quality control and quality assurance for our different cell types to be sure that we're producing the most high quality NAMs. So, in summary, what I've done today is to show how human cells and new approach methods based on iPSCs can speed drug discovery and drug development, and particularly relying on iPSC technology, which has advanced over time to create more advanced models. Now we're moving into a period of time where clinical trials on a dish will be more common and will help lead to a greater success rate of drug development and drug discovery for companies making those drugs, and importantly for the patients that need those drugs. So this is our biobank team. Our team is made up of very highly trained and highly skilled stem cell scientists who are expert in expanding iPSCs, which are a very difficult cell type to work with. Also, our biobank team has extensive experience in differentiation of iPSCs to various different cell types that I've shown in the webinar so far. So that concludes our first webinar for Greenstone Biosciences. If there are any questions, please put those questions into the Q&A and I will start answering those questions. I want to thank you for your time and for your attention this morning or this afternoon, depending on where you are. and I hope that you will tune in for our next webinar, which we'll send out an announcement about in the near future. So I'm going to go through some questions in the Q&A now. One anonymous question is, can you share information on what chronic lung disease iPSC biobank samples are available for researchers? So typically for disease cell lines, you can contact us through our info@greenstonebio.com email address or through our website that you see at the bottom, and we can provide you a list of those chronic lung disease patient samples that could be useful for your project. Another question says, can this replace animal studies needed for IND enabling studies? So as the regulatory paradigm changes, uh, there will be there will be a pathway for animal studies to replace, or for NAMs to replace some animal studies to enable IND studies with the FDA. There are examples in the case of cardiac toxicity testing where iPSC cardiac NAMs have been used in place of animal studies for IND applications that were successful with the FDA. There's a question from Priscilla Sato, an old colleague of mine. She asks if you would be able to order iPSCs and differentiate in-house. Yes, for academic institutions, actually, Greenstone Biosciences offers free iPSCs. There's a small restocking fee and some shipping fee, but yes, we do ship out iPSCs to academic institutions where you can perform differentiations on your own at your academic laboratory. There's a question from Kevin Costa. Uh, he mentions trials in France and Seattle that passed in animals but were toxic in patients. And he asks if there's any data from human NAMs that showed toxicity in human-based systems. So I'm not aware of any offhand of any data from human NAMs that have been used yet. That might be in the IND applications from drug makers that have utilized NAMs. For certain, it's possible using cardiac NAMs to detect arrhythmia liabilities of compounds that are known to cause arrhythmias. It's also possible using cardiac NAMs to detect specific ion channel block. This also can be done using neural NAMs and other cell type NAMs. Another question from an anonymous attendee is, are those patient-derived iPSC lines available for purchase? Uh, we don't typically sell the iPSC lines or license them out. Typically, we will partner with the customer to differentiate those cells in-house and then provide the differentiated cell types to the customer. There's another question, how the passing of cells affects the organoid culture. So iPSCs with passaging are remarkably stable in culture. So the passaging of iPSCs should not have an effect on organoid cultures. Uh, the organoid field is still pretty new, so I think there still needs to be a lot of validation and a lot of work done to determine how passaging of the cells can affect organoid function and organoid NAM testing. There's another question from Angelos. He would like me to comment on the size of each batch of iPSC-derived cardiomyocytes that can be produced for high-throughput screening. We make large batches. With each batch, we can generate around 50 million cardiomyocytes, which is typically sufficient number of cells to make significant NAMs and high throughput screening plates of monolayers. For the development of organoids, that requires a little bit higher number of cells, but we also can scale up to generate those numbers of cardiomyocytes for study. Another anonymous attendee asks, do you have isogenic controls for your cardiomyopathy models, or is this something we would need to work with you on to CRISPR engineer one by one? So we do have a small subset of iPSC lines where we do have a CRISPR engineered corrected cell line for a small portion of our cells. If there was a specific mutation that a customer needed to work with, we would work with you to CRISPR engineer that cell line one by one to make sure that we have high fidelity genome engineering to make sure that we have a proper isogenic control for study. Another question from Kalyani is, for cardiomyocyte differentiation, every passage of iPSC cells, I experience different success rates for successful differentiation. How to control these? So there is some inherent differentiation for each cardiomyocyte differentiation experiment. To control for the variability and differentiation, it's important to start with high fidelity iPSCs. It's important to ensure that the iPSCs are very pure and that there's not significant spontaneous differentiation, because that can impair and negatively impact the cardiomyocyte differentiation efficiency. So that would be one place to start. Another way to control is to have very dedicated and experienced personnel who are performing these experiments. And that's one of the strengths of the biobank team here at Greenstone, is that our scientists are very skilled and highly experienced in cardiac directed differentiation as well as differentiation to other cell types. Another question from Lucia. She says, thank you for the nice talk. Thank you for those nice words. Do you happen to be exploring the effects of microgravity in any of your current projects or cell line models? That's a great question. There is a lot of interest, and we do have contracts with NASA where we're studying the effects of microgravity on cardiac myocyte function and structure. So yes, that is a very interesting and very exciting aspect of our research, is studying the effects of microgravity. Joseph asks, please can you share how many clinical trial in the drug discovery area used iPSCs, FDA registered trial, and what is the current status? So I don't know the answer to that exactly, of how many clinical trials in a dish have been used and have been submitted to the FDA. My feeling is that it's not a great number at the moment. To perform a clinical trial in a dish really relies on an extensive biobank of iPSCs that can be used. I'm going to stop sharing now. Raised hand. So that concludes the webinar for today. Again, I want to thank you for your attention and for your very good questions. And I hope that you will consider partnering with Greenstone Biosciences for your NAM development and for de-risking compounds for your drug discovery and drug development projects. Thank you very much. This has been recorded, and we will send a copy of the recording to each of your emails. Thank you again, and have a great day.